Assalamu alaikum. Are you able to hear me? Okay. In the previous class, already we have finished the first module, right? In the first module, we have finished the SAS base and advanced. See, the next module is the clinical research and clinical data management. See, this is also important because it actually, so we are covering all the, I mean to say, the basic things about the clinical trials, right? What is a clinical trial exactly? What are the clinical trial organizations which are associated with the clinical trials, right? So all these things are covered, I mean to say, in this module. This is also very, very important for those who are actually not familiar with the clinical trials and clinical terminology. Okay, see, first of all, what is actually the clinical trial or clinical research, we can say, right? So, what is the clinical research? Because we have actually two parts in this clinical research and clinical data management. So, first of all, I will come to the definition. What is the clinical research? See, clinical research is nothing but first of all, it is a research done right on the human individuals, right? On human subjects. The first point is that it is a research, right, which is done on the human subjects, right, with the intention of what? With the intention to establish safety and efficacy. Understood? Listen carefully. This is a research. Clinical research is the research which is conducted, right, on human subjects, right, with the intention to establish safety and efficacy of new drug, safety and efficacy of new drugs or new, we can say, device or new procedure, right, or new actually surgical or diagnostic procedure which we are going to launch in the market for the first time. Understood? So this is the clinical research. Clinical research is nothing but it is a study, right, which involves human subjects, right? We are conducting the clinical research is done on the human subject, not on the animals. Understood? If you are doing on the animals, it is not called as clinical research, right? So that is known as a different word, right? But here the clinical research is nothing but where actually the research or the study is conducted on the human subject with the intention to study safety and efficacy of a new drug, right? New drug, or we can say new device, right? Or we can say new surgical or diagnostic procedure that we are going to launch, right? Newly for the first time in the market. Understood? That is nothing but clinical research. So this is how the clinical research, the clinical trial also, we can say clinical trial is also conducted on human subject, right? To for testing a drug, understood, or a device. So before actually anything is launched into the market, so what we are doing, first of all, we are testing it, right? So we are testing that actually the drug before actually it has been launched in the market, we have to test it on the human beings, right? Whether the drug, whether the new drug, right? Whether the new actually device, for example, sometimes we are launching the new devices, or for example, like blood pressure monitor is there, blood glucose monitor is there, right? So like this, different types of devices are there. So not only drug, maybe anything like which is newly launched, which is associated or related to human subjects, which are intended to be used on human beings. So such things, maybe it is a drug, maybe it is a device, maybe it is a surgical or diagnostic procedure, right? Which is newly going to be launched in the market, has to be tested on human beings. Understood? Has to be tested on the human beings. So that is nothing but the, I mean to say, the clinical research. So this is actually the clinical research, but and the clinical research is actually or clinical trial, we can say. So it involves actually so many steps. Understood? It involves so many steps. See, in one step, you cannot perform the clinical trial. I mean to say the clinical trial has to be performed, right, in several phases or several stages, we can say, right? Several phases or several stages are involved. Understood? In one step, you cannot perform, I mean to say, the clinical trial. The clinical trial is a very lengthy procedure, right? See, for example, one clinical trial, maybe it will take some time, 6 months to 1 year, or more than 1 year also. Understood? So, sometime it is involving what? More than 1 year also to perform the clinical research. So, clinical research is not like just in one day, the whole, I mean to say, uh, it will finish. It will take some time. so many months and so many years also, sometime 2 years also, 3 years, 5 years also. Some clinical trials are taking up to 5 years also, right, for the completion. So it is a very lengthy study compared to other studies, and it involves various phases. It involves various phases here. So we have actually different phases, but basically we have four phases in the clinical trial: phase 1, phase 2, phase 3, and phase 4. So these are the four phases in that the clinical trial is actually done. Understood? So usually the clinical trial is done in how many phases? In four phases. Understood? Not in single phase, it is done in phases. Phases means like parts, right? First part, second part, we can say like that. We have the phases. So like that, we have basically four phases, but some in the some trials we are counting one more fifth phase also, phase zero. So phase zero is not the I mean to say prominent phase. Some studies it is there, some studies is not there. But basically there are four phases. Basically four phases: phase one, phase two, phase three, and phase four. Understood? So these are the four phases in the clinical trial. Understood? So four phases are there in the clinical trial. So phase one, phase two, phase three, and phase four. Understood? So this is actually about the phases of the trial. And here, sometime actually I told you the phases are actually more than four also, right? Some studies we have phase zero also. Some studies we have phase zero also. Phase zero is not like a it is the main phase. It is actually sometime present, sometime not present. Often in most of these studies, right, phase zero is not present. Phase zero is not present. In some exceptional cases, phase zero is present. So we are not counting the phase zero. Actually, phase one to phase four are the four phases. Phase zero is sometime present in some studies only, right? As per the requirement, phase zero is added. And before the four phases, there are preclinical studies. Preclinical studies are nothing but before starting the trial. See, just listen carefully. The preclinical studies are not in the phases. It doesn't count in the phases. These are actually the, I mean to say, preclinical studies are done before starting the clinical trial. So why we are doing the preclinical studies before starting the clinical trial? Because clinical trial, just like I told you, will be conducted on human subjects. See, the people who are participating in the trial are known as subjects. Just remember this term. This term is very important because from time to time you are going to hear this. I mean to say the term subject. See, subject means people or the volunteers. Subject means the people or the volunteers who are participating in the trial are known as subjects. Understood? The people or the participants or the volunteers who are participating in the trial are known as subjects. Understood? And here all are human subjects only in the trial. In the trial, actually, human subjects are used. But directly you cannot test any drug on human subject. For example, you are going to test the drug on human beings. So before that, we don't know how the drug is acting, whether the drug is safe or not. See, actually, the main intention of that clinical trial, just now I given the definition of the clinical research. So it is the study done on the human subjects to establish the safety and efficacy. The two things I have told you there, if you remember, just now I told you the definition: safety and efficacy. These two terms I have used. See, this is very important. Any drug which is going to be used on human beings, there should be two things. One is what? It should be safe to use. It should be safe to use, and it should be showing some efficacy also, effect also. See, for example, water is there. Water is safe to use, right? Water, for example, which we are drinking, is safe to use, but it is not a drug because it's not having any efficacy. So it should be having safety also. It should be having efficacy also. For example, take a poison. Take any poison, for example, every poison will have some efficacy. Yes or no? See, any poison if you take, maybe in small amounts, it can be used as a for treating some I mean to say disease also. Some poisons are actually can be used for treatment also, but they are not safe. The poisons are not safe to use. So we cannot say them the drug. So for to be called as a drug, it should be safe. It should be showing efficacy. Both the things should be present in the drug. See, water is there. Water is safe to use, but it's not a drug. Why? It's not showing any efficacy. Any poison is there. Poison is actually showing some efficacy in some cases, but it is not safe to use. So we cannot call we can't call the poison as a drug. So to call a substance a drug, it should be safe. It should be showing efficacy. Now we don't know any substance whether it is safe to use in human beings or not. So what we will do? Directly we cannot test any chemical substance on human beings. Suddenly something happened. The person on whom we are testing the drug, maybe they will die. Yes or no? Because drugs means they are chemicals. Drugs means most of the drugs are chemicals and they are dangerous. The drugs are not actually like a normal substances. They are harmful chemicals. So we cannot take risk for doing the clinical research or clinical trial directly if you are testing the drug on human beings at the first means starting only. What will happen? Maybe that drug is not a drug. That maybe that substance is very dangerous. Maybe that substance. What happened to the COVID vaccine? See what happened to the COVID vaccine that has not been tested on human beings, not broadly tested. What is the result now? What is the problem now? All the COVID vaccines, most of them are proved to be very dangerous, very fatal. So many people they are dying due to heart attack and so many other problems who have taken the COVID. Understood? COVID vaccine. So in this way, if you are not testing the drug properly, what happened? Directly you cannot. I mean to say the human beings they cannot use it. They cannot use that chemical substance. They may be dangerous, right? They may be dangerous. So first of all, they have to be tested. And before testing on human beings, for knowing that drug is actually not a poison, not causing any very dangerous side effects, first we are testing on non-human subjects, means non-human targets. We are using the drug, like for example, we are using the drugs on animals. Understood? We are using the drug on animals, or we are using the drug on, for example, like some tissues which are extracted from animals, sorry, from human beings also. Like for example, you are taking some tissues from liver, some tissues from the any from our body also you can take. So directly instead of testing on human beings first time, we are testing on non-human subjects, like we are testing on animals, right? Most of the drugs are tested on mice, right? Some drugs are tested on rabbits. Some drugs are tested on guinea pigs and monkeys. So they are tested first on animals, or they may be tested in the lab also on non-animal. Nowadays, actually, the cruelty to animals, like there are so many rules and regulation laws are there in India also. Maneka Gandhi, one actually activist, she is actually condemning this use of animals in the research. So sometimes we are using the non-animals also, means no animals, no human beings are used for testing the drug, like tissues taken from the culture, right? Like human tissues or animal tissues. We are taking on the tissues, we are testing the drug. So these all things which we are doing, right? The testing the drug on non-human subjects, either on animals or on tissues, is known as preclinical studies. What do you call this one? Preclinical studies for non-human subjects. So why we are doing these preclinical studies? Preclinical studies are done to study the toxicity of the drug, especially the toxicity of drug, and efficacy also, means whether the drug is showing any action or not. See, simply before testing the drug on human beings, every clinical trial is a very huge setup. Clinical trial is a very huge setup. Actually, it is having millions, multi-million dollar projects. Sometimes millions of dollars are used in the clinical trial. So, for example, here we don't know whether drug is going to act or not. First of all, the chemical is a drug itself or not. How we can say this chemical is going to act, this chemical is having some good effect? How we can say that this chemical can be used? How we can say this chemical can be used as a drug? Understood? We don't know, first of all. We don't know anything about this chemical, whether it can be used as a drug or not, first case. And whether it is safe, maybe it's a poison, maybe it's toxic. So we are testing on the animals or non-human subjects. Understood? So this will actually rule out any problem. So we don't want to risk the human life. We don't want to risk the human life. So that is actually known as preclinical studies that are done on the non-human subject to know the toxicity of the drug, whether it is poison or not, or whether it is showing any effect or not, whether we have to carry on the research or not. See, unnecessarily wasting the money on research. First, we will actually confirm on the non-human subjects that this actually substance is a potential target, potential actually substance which can be used as a drug. It is not having any toxicity. We want to confirm by using on animals, and we want to confirm that actually this is showing some efficacy, some type of like action, pharmaceutical, I mean to say pharmacological action. It is showing that actually can be used on human beings. So it is showing some positive results on animals. Then we can say, okay, this drug is actually not toxic on the animals and have some, I mean to say, the positive effect. That means it is showing some good effect on the subject. So that can be now further can be tested on human beings. To make sure this, we are doing the preclinical studies, and here actually the amount of drug used for testing is unrestricted. So there is no such fixed dose. For example, for every drug there is a dose. Dose is nothing but the amount of drug that can be used right safely by any person. Understood? For example, paracetamol, if you take, right, you are taking Calpol, you are taking Panadol, for example, any drug containing paracetamol, we are usually taking 500 milligrams. How much we are taking? 500 milligram to 1000 milligram is the dose. So like that, for preclinical studies, there is no fixed dose, because here we don't know anything about the drug. We don't know whether this drug is safe or not, how much has to be given to the patient. Nothing we know about the drug. So freely we are giving the drug. How much drug is to be given is not fixed, not fixed in the preclinical studies. And this is carried out by a scientist, right? A scientific researcher will carry out these what? Preclinical studies. And here the human beings are not involved. Here no human beings are involved, right? Here, for example, we are using the non-human subjects. Sometimes the cells, I told you, the tissues extracted, the human tissues are used, understood, to carry out the research, I mean to say. See, now coming to the phases of the trial, I'm telling you, see, actually the phases of the trial start from phase 1, but here the phase 0 also given, but phase 0 is not the prominent phase. In most of these studies, we don't have phase 0. Phase 0 doesn't exist in most of these studies. What is the phase 0? See, phase 0 is actually we are doing on the human subject, very small number of people. Very small number of people we are recruiting in the trial. How many people? 10 people. Only 10 people we are recruiting in the trial for phase 0. For what purpose we are doing the phase 0? The primary goal is what? Pharmacokinetics. Understood? To check the bioavailability and half-life of the drug. See, these terms you are not maybe familiar if you are from medical background, right? You can understand these terms. What is pharmacokinetics? What is bioavailability of the drug? See, pharmacokinetics in short, if you want to know, is nothing but what the how the body is responding, how the body is responding to the drug is known as pharmacokinetics. Just just actually keep in mind, pharmacokinetic means how the body is responding to the drug, how the body is actually absorb, how the drug is absorbed, how the drug is absorbed in the body, how the drug is actually distributed in the blood, how the drug is actually metabolized in the blood by the kidneys or by the lungs, right? Sorry, by the liver, not lungs, by the liver and the kidneys, how they are metabolizing the drug. Metabolizing the drug means what? See, whatever the drug you are taking in the whenever you are eating the tablet, something like paracetamol, whatever, first of all, it will go into the stomach. In the stomach, it will dissolve, right? Then it will be absorbed in the blood. It will go to the blood. From the blood, it will go to the different parts of the body where the drug will act. And after the action is over, the drug will be metabolized. Drug will be metabolized means the drug will be destroyed. Understood? The drug molecule will be broken down into small molecules. That is known as metabolism. So all these things are done by the body itself, right? That is known as pharmacokinetics. So to study the pharmacokinetics, how the drug actually is responded by the body, how the body is taking the drug, means the body is actually responding to the drugs properly or not. We are doing what? Pharmacokinetic studies we are doing. Understood? The pharmacokinetic studies we are doing here, and also the bioavailability of the drug and half-life of the drug means what? See how much drug is available in the blood. See, you are taking drug 500 milligram. Maybe out of 500 milligram, only 200 milligram will be go into the blood. Rest of the 300 milligram will be directly go out without going into the blood. So how much drug is available in the blood is nothing but bioavailability. After taking the drug, how much drug is absorbed into the blood is nothing but bioavailability, and half life is nothing but how much drug is actually metabolized, right? For the half of the drug, how much time is taken? How much time is taken to metabolize the drug into half? For example, you are taking 500 milligram, from 500 milligram to 250 milligram. See, drug from 500 milligram, it is actually metabolized to 250 milligram. How much time is taken for that is known as half life. So anyhow, so these are the studies which are not actually directly concerned with our main goal, but we want to study. We want to see how the drug is how the drug is behaving in the body. For that, we are doing phase 0, and here we are using the subtherapeutic dose. How much we are using here? Subtherapeutic dose means very small amount of dose is used. For example, the therapeutic dose is nothing but the dose at which the treatment of the patient is done. The dose which is actually causing some effect on the human body is known as therapeutic dose. For example, for paracetamol, just like I told you, which is used for fever and headache, Panadol, Calpol, right? These are the brands of paracetamol. They are used for what purpose? They are used for fever and headache. What a therapeutic dose of paracetamol? 500 milligram. For an adult, how many milligram of paracetamol should be taken? 500 milligram of paracetamol should be taken. Understood? So if you are giving the dose less than 500 milligram, like let us consider we are taking 100 milligram only. If you are using only 100 milligram of paracetamol, it is called as subtherapeutic dose. Are you getting me? It is known as subtherapeutic dose of paracetamol because 500 milligram is the main therapeutic dose. Less than that, which we are using in the study, is called as subtherapeutic dose. So in phase 0, we are not using much drug. We are using very small quantity of drug, that is known as subtherapeutic dose. And this from here onwards, this is actually performed by clinical researcher. All this actually the work from phase 0 to phase 3, right, is done by the, I mean to say, clinical researcher is done. Understood? The clinical researcher is different, scientist is different. See, scientist is a normal person who may not have the medical background, but clinical researcher should be having medical background. Understood? He should have the clinical domain knowledge. That is the clinical researcher. And here the number of people are very less, I told you. Small number of people are taken, only 10 people. Why in phase 0 we are taking very small amount of people, 10 people only we are taking in the trial? The reason is that if you take more number of people, we don't know how the drug is going to behave. Understood? We don't know how the drug is going to behave on the human beings. Till we don't know. Still we don't know because we have started now in the phase 0, testing on the human beings. We don't know whether the drug is actually safe for the human beings or not safe for the human beings. Okay, the drug is already tested on animals before in the preclinical studies, but still in the phase 0 we cannot use many volunteers or many subjects because first time we are using on the human beings, so you cannot take the risk again. So we are taking only 10 people only. 10 people only we are taking in the trial. So such actually studies also known as pilot studies. What we call this type of studies? Pilot studies, where actually very small number of subjects or volunteers are involved. Understood? And here this phase is not important. Phase 0 is, I told you before also, usually the phase 0 is not done. The phases starting from not phase 0, actually starting from where I told you just now, it is starting from phase 1, phase 2, phase 3, phase 4. Phase 0 doesn't exist for most of the studies. For most of the studies, there is no phase 0. Clear? Now coming to phase 1. See, phase 1 is actually, just 1 minute, hold on, 1 minute. Okay, so I was talking about the phase 1, right? See, phase 1 is the first, actually the first phase starting from phase 1 only. The first phase of the trial is nothing but phase 1. So what is the phase 1? Why the phase 1 is done? See, the phase 1 is done to establish the safety of the drug. Listen carefully. See, all the phases, there is a one purpose. One goal is there. One goal is there for every study. See, every phase is a separate study. Every phase is a separate study, we can say. See, phase 1 is done for what? To establish what? Safety of the drug. See, whether the drug is safe to use in human beings or not, we want to establish, we want to confirm. For that, we are doing the phase 1 studies. See, what we are taking in the phase 1 studies? In the phase 1 studies, we are recruiting healthy volunteers. Whom we are recruiting here? Healthy people are only taken as a volunteer in the phase 1 studies. We cannot take the diseased people. Why? Now you can ask me why we are using the drug on healthy people. See, healthy people, we don't want to, they don't have any problem. So why we are using the drug on healthy people? The reason is that we want to check whether the drug is safe or not. See, if you are using on diseased people, what happened? Already the diseased people are actually they are very sensitive. They are having actually the compromised body. Their body is weak. Their body is weak. Actually, we cannot test a drug directly for the first time on diseased people. So we cannot take the risk. So maybe, for example, one person is weak, some drugs are very strong, maybe it will cause very dangerous effect on the weak person. So we don't want to take the risk. That's why we want to test the drug for the first time. That is the phase 1 is the first time, right? Phase 0 is not the first time. Phase 1 is the first time. So first time we want to test the drug on healthy people only. So maybe if it is the drug is actually dangerous also, the healthy people will get the less, I mean to say the damage. Understood? The less damage will occur, less side effects we can say, or less actually means like problem will be faced by the healthy people compared to the diseased people. So that is the reason here we want to use the healthy people here, because the healthy people will actually respond if any dangerous event also happened, or there's something happen also, the healthy people can tolerate. Healthy people can tolerate the that effect. So that's why we are using the healthy volunteers in the phase 1 studies. And here the dose is actually usually what? Subtherapeutic dose only. What is the meaning of subtherapeutic dose? Subtherapeutic dose, I told you, the therapeutic dose of paracetamol is how much? 500 milligram. If I am using 100 milligram of paracetamol in a clinical trial, it is known as what? Subtherapeutic dose. See, what is the therapeutic dose of paracetamol? 500 milligram. If I use only in a study, I'm going to use how much? I'm going to use only 100 milligram of paracetamol. So what do you call this 100 milligram of paracetamol is called as subtherapeutic dose. So in the phase 1 trials, we are going to use only, I mean to say, subtherapeutic doses only. Why still subtherapeutic doses? Why not the full dose? Why not the full dose we are using here? The reason is that still we don't know. Still we don't know the drug is safe or not. Maybe the drug is dangerous. Maybe the drug will cause some serious side effect. Maybe some drug will be actually harmful. So we cannot take the risk. We cannot take the risk. If we use more drug, definitely the harm or the side effect or the danger will be more. Yes or no? See, for example, if you're taking small amount of anything, the more problem, more actually damage or more harm it will do. Or if you take more amount of anything, it will do the more harm or more damage. Definitely, large quantity of any substance if you consume, if it is a poison, then definitely that person will die. If the person consume only small amount of any poison, maybe he will not die. Maybe he will not die. So that's why we don't want to take the risk here. The drug, maybe we don't know whether it is a good or bad. That's why we are taking subtherapeutic doses only in phase 1 also. But what we are doing in each step, we are increasing the dose slowly. What we are doing here? We are ascending in ascending order, ascending doses. The drug is slowly increase. For example, first day we are giving how much? 100 milligram of paracetamol. Second day we are giving what? 150 milligram of paracetamol. Again, we are giving now 200 milligram of paracetamol. Slowly, we are increasing the dose so that suddenly something happen, so we can control. We should know how much dose maximum the body can tolerate. Understood? So slowly, we are increasing the dose. We are starting with a small dose. Then what we are doing in each step, the amount of drug, what we are doing, the amount of drug is slowly increase from 100 milligram to 150 milligram. Then the next from 150 milligram to 200 milligram, right? Again from 200 milligram to what? 250 milligram. Slowly, the drug actually, what we are doing, we are increasing the strength, increasing the dose. Understood? Slowly, we are increasing the dose in every step. That is when ascending doses are taken. So this is how finally we want to know maximum dose. Maximum dose we want to fix. For example, I'm starting with 100 milligram. I'm giving 150 milligram, 200 milligram, 250 milligram, 300 milligram, 350 milligram, up to 500 milligram, or maybe up to 1000 milligram also. I will increase it. Slowly I will increase. Then I will give up to 500 milligram, 550, 600, 700. After reaching 1000 milligram of paracetamol, because paracetamol can be taken 1000 milligram also. Up to 1000 milligram it can be taken. So after reaching 1000 milligram, then again, if we increase the dose further, it will cause the very harmful effect. So that is the maximum dose. See, 1000 milligram is the maximum dose up to which we can give to a patient. More than actually if we use paracetamol. See, paracetamol is very common drug. Most of the people, even the small infant, kids, children's, they are using paracetamol. But that also having some limit. See, that paracetamol has to be given within a limit, within a safety limit. Beyond that safety limit, if you take paracetamol also, it will become poison. For example, 1000 milligram is the last maximum dose which can be given for the paracetamol. If you give more than 1000 milligram, it is dangerous. Paracetamol will cause liver damage. What it will cause? It will cause here what? Liver damage. That's why the maximum dose, how much can be given is actually we are finding through phase 1. So phase 1 study will help in deciding the dose of the drug. The maximum safe dose, I mean to say. The maximum safe dose we can actually we can find out by using phase 1 studies. So phase 1 studies are used for what purpose? Phase 1 studies are used for safety studies to establish the safety, whether the drug is safe to use, right, on human beings or not. And next thing is what? We are giving the subtherapeutic doses and slowly we are increasing the doses until, right, the maximum dose beyond which we cannot give the drug. So up to that dose, maximum safe dose, we are going to find out in the phase 1 studies. And healthy volunteers are used. What we are using here? Healthy volunteers. And how many people? And it is done by the clinical researcher. And how many people are involved in the phase 1 studies? We are taking 20 to 100 people. How many people? 20 to 100 people are actually usually involved in the phase 1 studies, not more than 100. So this is also small group only, not very big amount of people we are taking here. Why? Why we are not taking 1000 people? Why we are not taking 2000, 3000 people? We can take, no. But in the phase 1 studies, we are not taking more number of people. We are taking less than 1000 people, less than 500, less than 300 only. Not less than 500, less than 300 people only we are taking. Why? Why this many less people we are taking here? We can take more people also, 1000 and more than 1000 also. But we don't know the drug is safe or not. Understood? Because we don't know still the drug is safe to use or not. So the safety profile of the drug is not established. Still we don't know the drug is safe. That's why we cannot take the risk. See, more number of people we are recruiting in the trial. Suddenly one serious side effect happened, like COVID-19 is there. What happened? For COVID vaccine, I mean to say. Suddenly what happened? We use the drug on so many people. Thousands of people use the COVID vaccine. What happened? So many people are affected. So we don't want to take risk. That's why the drug is used on some less number of people, only 100 to 200. Less means 100 to 200. We are not taking large number of people because maybe we don't know still. In the phase 1, we are going to see the safety. We don't know whether the drug is safe or not. After phase 1 studies only, we are confirmed that the drug is safe. Understood? After finishing the phase 1 studies only, we can confirm that the drug is 100% safe now. It can be used on other human being also. Now we can increase the number of people from phase 2 onwards. From phase 2 onwards, we can take more number of people. Understood? So here 100 people only taken here. Sorry, in the phase 1 we are taking only 100 people, not 300 people. Less than 100 people only we are taking, not 300. In the phase 1 we are taking 20 to 100 people only. 20 to 100 people only we are recruiting in the phase 1, not 300, even less than 100. Less than 100 people are taken in the phase 1 studies, and normal healthy volunteers are taken. And here success rate of the phase 1 study is 70%. 70% is the success rate for the phase 1. What does it mean? See, 70% of the drug will pass. 70% of the drugs will pass this phase 1 studies. 30% will fail. 30% will fail in the phase 1 studies. So here this is used to check the safety of the drug before using the drug, right? We want to check the safety of the drug. For that purpose, the phase 1 studies are done. So here the phase 1 studies are over. So we'll see tomorrow, right? Other phases also in detail. So meanwhile, the time is over. I will stop here. We'll continue the next class, right? So today we have finished the phases, right? I mean to say, phase 1 we have finished, right? So tomorrow, inshallah, we'll see the phase 2, phase 3, phase 4, right? How the different phases of the trials, right? So that we will see in detail, inshallah. So I will stop here. We'll continue in the next class. Okay, Allah Hafiz. Assalamu alaikum.